The movement of plants is nowhere more obvious than in the infinitely diverse mechanisms used to eject the spores. In a large group of ferns known as the leptosporangiate ferns, spores are ejected by a mechanism that is aptly described as a cavitation catapult. The spores are kept in sporangia held in small clusters, the sori, under the fern leaf. Close examination of the sporangia, when exposed to air, reveals a slow opening of the spherical capsule, followed by a rapid closure during which the spores are ejected. To understand how the spores are ejected, we must look more closely at the structure of the sporangium. The sporangium is a thin spherical capsule that encloses the spores. A row of specialized cells sits to one side of the sporangium. The cells of the annulus have thickened inner and radial walls forming a reinforced U-shaped structure, while the lateral walls are rather thin. When the cell loses water to the environment, its volume must decrease. This is accomplished by bringing the radial walls closer to each other and by collapsing the lateral walls inwardly. This clip shows the intricate relation between the decreasing cell volume and the curving of the annulus. Over a period of a few seconds, the annulus reverses nearly completely its curvature in response to the ever declining cellular volumes due to evaporation. The strong curvature that develops along the annulus must be supported by a balancing force. This force is provided by the suction force, or negative pressure, that the cell content exerts on the cell walls. The continually decreasing cellular volume and bending of the walls invariably led to more negative pressures within the cells of the annulus. Beyond a certain point, the pressure within the annulus cells is so high that their content cavitates. Various estimates indicate that a negative pressure of 100 to 200 atmosphere is present within the cell at the time of cavitation. Cavitation effectively releases the suction force that was holding the annulus in its bent configuration. The annulus thus returns quickly to its original configuration, ejecting the spores in the process after this nucleation of bubbles. However, the greatest refinement comes into play only after cavitation. The spores would not be ejected if the sporangium simply sped up to its closed geometry. Its returning motion must be arrested abruptly for the spores to be ejected. The same problem was faced by the designers of the first catapults. In catapults, a crossbar stops the arm in its course, thus allowing the projectile to be launched. This crossbar is strikingly missing in the fern sporangium. We have found that the sponge-like organization of the annulus walls allows for the two very different closing timescales that serve the same purpose. The fast timescale is inertial and is observed in any elastic material that is suddenly released from a deformed geometry. The second timescale comes from the need for water to flow across the annulus wall to adapt to the rapidly changing geometry. The flow of water within the tiny pores of the wall is impeded by viscosity, leading to an abrupt arrest of the sporangium motion. It is remarkable that a dozen cells have evolved to fulfill all the critical functions of a catapult.